Our next speaker is Lonneke Scheffer, presenting CompAIRR.
Hello, everyone. As Justin said, my name is Lonneke Scheffer. I'm a PhD student at the University of Oslo, and I'm presenting CompAIRR, which is a tool for efficient computation of overlap between immune repertoires and also allowing non-exact sequence matching. So there are two main motivators for us developing the CompAIRR. So first of all, we wanted to have a fast and efficient algorithm for immune repertoire overlap, because there are many AIRR applications that internally are needing to calculate AIRR overlap, such as when we want to calculate distance metrics between immune repertoires, which is something that can, for example, be done by the tools shown on screen, VDJ tools, immuneARC, and immuneRef. But as our sequencing technologies improve, we're sequencing larger and larger data sets. So the software tools that we used in the past that might not be able to keep up with these larger and larger data sets. So we need to have software that is able to scale up to these larger data set sizes. And secondly, we wanted to allow sequence mismatches when we compare or when we look for the number of matches between immune repertoires, because as we know, there are only very few public sequences in immune repertoires, but similar CDR3 sequences might imply similar antigen binding capabilities. So when we calculate how many matching sequences there are across repertoires, it might also be interesting to allow a little bit of mismatches in these sequences. And this is not, this functionality is not provided by any of these existing tools. So therefore, we created CompAIRR, a fast and memory efficient tool for AIRR overlap. So as I said, CompAIRR can work either with exact sequence matches or allow a small number of substitutions or insertions and deletions. We can choose to include or exclude V and J gene information and sequence frequency information. And internally, we are using fast and memory efficient data structures such as Bloom filter and Zobrist hashing to speed up the lookup of matches across repertoires. So as an output, CompAIRR generates an overlap matrix, which can contain many different basic raw statistics, but we can also directly output a Morishita-Horn or Jacquard distance matrix of all against all repertoires. And then we can also export the exact sequences that were found to match across repertoires to an AIRR format output file, which can be used for further analyses with other tools. So how fast is CompAIRR then? We benchmark CompAIRR to the three previously mentioned tools, immuneARC, immuneRef, and VDJ tools. So we used all these tools to calculate the number of overlapping sequences between different sets of repertoires, where on the small data set size, we have 10 repertoires and going all the way up to 10,000 repertoires, comparing all against all repertoires in the set. And here I'm showing the running time of the different tools. And I'm including two versions of CompAIRR, one with a single thread and one using the multi-threading option. And so the fastest version of CompAIRR with multi-threading took only 17 minutes on this largest data set to compare all against all, whereas immuneARC took 10 days, immuneRef took 23 days, and VDJ tools did not complete due to memory limitations. So CompAIRR is also more memory efficient than these other tools, and it used at most 1/3 of the memory compared to the other tools. So in the previous two figures, I only showed the results for exact sequence matching. Yeah, as I said, CompAIRR can also do non-exact sequence matching, which is a bit more computationally expensive because there's a lot more different matches that need to be looked up. But there we can see really that the multi-threading option provides some benefit in reducing the running time. And for all of the figures that I've shown here, the raw data and scripts for reproducing the benchmarking can be found also on GitHub, which is also linked from our preprint. So apart from the repertoire overlap functionality, CompAIRR also has some other use cases that we included. So we can create a sequence presence matrix, which is where if we want to compare repertoires to a set of sequences and know for each individual sequence whether it was present or absent in the repertoire and just get one big table as an output. And here we also have provide the non-exact sequence matching options. And finally, we can do single linkage clustering of a set of sequences. So here it's just every time that the sequences are a given number of substitutions away, they are put together in a cluster. And this way we can grow out the clusters larger and larger. So CompAIRR is not trying to replace the previously mentioned tools, but rather it's trying to speed up this core component, because these other suites that I mentioned, immuneARC, immuneRef, and VDJ tools, they have a lot of other functionalities as well. And we are only speeding up this core functionality of repertoire overlap. So we showed that CompAIRR can be integrated in another tool. And here as an example, I'm using the machine learning platform immuneML, where we integrated CompAIRR in two different ways. One way we integrated it as with the repertoire overlap functionality to create a Morishita-Horn distance matrix, which can be used in combination with the KNN classifier in immuneML. And we also used the sequence presence functionality to speed up a method from Emerson and colleagues that we reproduced inside of immuneML. And then finally, I would like to thank everyone that made this project possible, and in particular Torbjörn Rognes, who developed CompAIRR, and Gersetel Sandve and Victor Greif, who supervised the project, as well as all other people and funding that made it possible. So thank you very much.
Great. Any questions for Lonneke?
I have one question, but
Yeah.
first, thank you for outputting the AIRR format. Let's get you on the website. That's fantastic. Are you able to account for ambiguous nucleotides in your inexact matching by any chance?
You mean a sequence that, yeah, no, I don't. I think we actually have an option for including an extended alphabet, but then the ambiguous nucleotide will not necessarily be matched to all the possible nucleotides that it could be, only to the character, the exact same character. But it is a good suggestion that we could maybe include in a future version.
Thank you. Nice tool.
Nice talk. What distance metric are you using for grouping your sequences?
So here we're just, you can use the same non-exact sequence matching metrics to configure CompAIRR. So you can either use a number of substitutions or insertions and deletions for making the clusters.
Just one comment. Just one quick comment from the Zoom chat. Very exciting improvements, and I love the name of your tool, from Nina.
Thank you. I came up with it myself.
Great. Thanks
I'm
very much.
a very punny person.
Thanks very much, Lonneke.